Today my topic is mechanism of translocation through sieve tubes. The mechanism to explain translocation of solute through sieve tubes were understood by extensive studies in the concentration of substances in living sieve tubes. The rate of translocation through them and the effect of temperature and environment and factors. The translocation of solutes in the sieve tubes of plants, these are could be explained by two theories. First one is the cytoplasmic streaming theory. Cytoplasmic streaming theory. This theory was proposed by De Vries and De Vries and later supported by uh, Curtis in 1935, and according to this theory, solutes are carried by combination of diffusion and streaming of cytoplasm. Diffusion this takes place from sieve tubes through pores of cross walls within the sieve tube. Substances would be transported downwards or upward along the cytoplasmic stream. The main objections of this theory: the rate of streaming of protoplasm is much less than the rates at which solutes move through phloem. The rate of cytoplasmic streaming is only 1 to 3 centimeters per hour. However, the rate of streaming is identified as 100 to 150 centimeters per hour. Hence, the mass flow of solutes. So, this is the translocation to by this cytoplasmic streaming. Uh, this one this one is sieve tube, and this one this B is companion cells, this A is sieve tube. So these are big arrows, these are cytoplasmic streaming, and these small arrows indicate solute flow. So these are big arrows. These the cytoplasmic streaming. These very small arrow marks are here. These are indicate solute flow. So this is translocation. So the mass flow of solutes. The solutes to long distance is not possible through cytoplasmic streaming. However, in roots, cortex, and parenchyma cells of leaves, solutes, these are translocated from one cell to another cell through cytoplasmic streaming. So, older sieve tubes, these have no streaming protoplasm, and only young sieve tubes have streaming protoplasm. So next theory, first one is this one. So in the first one, cytoplasmic streaming theory, these are main objection of this theory is the rate of streaming of protoplasm. So this is much less than the rates at which solutes move through phloem. So rate of cytoplasmic streaming, it is only 1 to 3 centimeters. So the rate of streaming is identified as 100 to 150 centimeters. This mass flow solutes to long distance is not possible through cytoplasmic streaming. However, in root cortex and parenchyma cells, and living leaves cells of leaves solutes can be translocated from one cell to another through cytoplasmic streaming. So, older sieve tubes, these have no streaming protoplasm. Only young sieve tubes have streaming protoplasm. So, second theory is the pressure flow hypothesis. Pressure flow. This is a physical model to explain translocation of solutes by pressure flow. This is hypothesis proposed by Munch. So Munch, he is a German scientist, put forward pressure flow or mass flow hypothesis. Munch explained his theory using a physical model. This model consists of two osmometers, A and B. This is the A, low concentrated solutions. B is the more concentrated solutions. So the Munch consists of two osmometers A and B with semi-permeable membrane connected by means of narrow tube. This is C. So this one is the narrow tube. This is semi-permeable membrane. So the C and these are kept separated in two different systems with water connected by a narrow tube. This is shown in this picture. So the first osmometer A, it is the filled with low concentrated solution, and the second osmometer, this one is filled with higher concentrated solution. More water enters by osmosis into the second osmometer B. So the first osmometer, as it has more concentrated solution and less water enters into the first osmometer A. As the water potential difference between the external system and that of A is very less, the more water enters B than A, more positive hydrostatic pressure is developed in B, and as the two osmometers are connected by the narrow tube C, the positive hydrostatic pressure is transferred to A from B, and water moves out of A to external medium due to the relative increase of free energy. Thus, water moves from B to A as a mass flow and as a mass flow, and along with the water solutes dissolved in the water also move with water. These solutes are shown as dark spots in the osmometer. A can expand with inflow of water. The pressure in it will not increase and water will not move out. The long distance transport in plants could be easily understood by comparing with the physical model. The osmometer B can compare to source in plants which synthesis food materials, and the osmometer A can be compared to sink which utilize food as sugars as synthesized in leaves. The concentration of leaf cells will increase and is comparable to the osmometer B, while the roots, flowers, fruits, these are unstill the materials and the concentration in these cells is less, which is comparable to the osmometer A. So, in plant cells, the materials are used by respiration, and some of the sugars also. These are converted to the reserve materials like starch, further reducing the concentration and the osmotic potential. These becomes more negative, and the solute concentrations in zinc is always less, resulting in the continuous translocation of solutes from source to sink. The incomplete lines in this picture indicates the increase the volume. These are comparable to the growing fruits. And the narrow tube connecting the two osmometers is comparable to the simplest system in the plants, and the tube connecting the two containers outside the osmometers to the apoplast system. So, apoplast system, cell walls, intercellular spaces, water with solutes in xylem. So, the main differences between the physical model and plants is that the physical model equilibrium is established after some time, where as in plants, equilibrium is not established between source and sink, as source always synthesis food materials and sink always utilizes the food materials are converted to insoluble from like starch, and the food materials synthesized or translocated continuously from source to sink in a positive manner. So this is the two methods. First one, this one is the pressure flow hypothesis proposed. This was Munch, and he is the German scientist. This forward pressure flow or mass flow hypothesis. This Munch explained this theory using a physical model. This model consists of two osmometers, A and B. Second one is mechanism, cytoplasmic streaming theory. In this theory proposed by Davries and later supported by Cortes in 1935. According to this theory, solutes are carried by a combination of diffusion and streaming of cytoplasm. So, diffusion takes place from sieve tubes through pores of the cross walls. Within the sieve tubes, substances would be transported downward or upward along the cytoplasmic stream. So, this mechanism to explain translocation of solutes through sieve tubes. These are so these substances and living sieve tubes and this mechanism understood by extensive studies in the concentration of substances in living tube, the rate of translocation through them and the effect, the temperature and environmental factors. The translocation of solutes in sieve tubes of plants could be explained by two theories. First one is the cytoplasmic theory, second one is the hypothesis physical model theory. These are two theories. These are two theories. So evidence for mass flow or pressure flow in plants. So the favorable conditions created in the physical model for pressure flow also exist in plants. The concentration gradients in the two osmometers of the physical model also occur in plants. The concentration gradient exists between source and sink. Mm, the photosynthesizing leaves prepare sugars and this have higher concentration of solutes, while the organs like roots mm, utilizing of food materials have less concentration. Further, the concentration of solutes in the transport system from stem tip to the roots gradually decreases. So the water potential is very less in source. In sinks, the value is relatively more. Thus, the more water enters source, increasing the pressure in them. So the narrow tube connecting the two osmometers also exist in plants. So in plants, sieve tubes acts as functional intermediary system, but not as a continuous narrow tube as in the physical model. So the sieve tubes possess sieve plates with minute pores. So the this doubt arises whether the solutes can move through the minute pores. The several evidences exist to show the translocation of solutes from source to sink through pressure flow. So it is the noticed when the slit is made in the reproductive parts of borasus palm trees, and uh, with a knife around 10 liters of fluid oozes per day. So and more than 10% of which consists of sucrose. So it is the already learned that the fluid flowing through phloem is rich in sucrose. So the fluid flowers from the reproductive parts of the borasus palm is synthesized in the borasus palm is synthesized in the leaves and translocated tree. This clearly indicates the fluid flowering through sieve tubes have uh, pressure. The cells synthesizing sugars in leaves are connected through the plasmadesmata with sieve tubes as observed under electron microscope studies. So another evidence for the translocation of solutes through sieve tubes is existence of apoplast and symplast systems in plants. So sieve plates at the ends. The are connected with one another from the one end to other source to sink as continuous system called as symplast system. So through xylem vessels are nearer to the sieve tubes, they are completely separated from xylem vessels. These are en- enveloped by the moist cell walls. Like sieve tubes, xylem vessels also form continuous system from source to sink, and the whole system which xylem vessels and the other cell wall with moisture together called as apoplast system. So this apoplast and symplast will be uh, these are okay, I will explain. Symplast and apoplast I will explain uh, the in the physical model of Munch.